How many steps in glycolysis are irreversible? 3. Okay. And how many? So let's pick one example. The first enzyme that is irreversible in glycolysis. Which one is it? Hexokinase. So let's look at hexokinase. And so just remind everyone, what does hexokinase do? Right? Glucose to G6P. So that's the first step. What it means is if you have glucose in the blood, grab it into the cell and trap it. We have looked at the muscle cells, right? How the insulin secretion made the membranes containing the glucose transporter fuse with the plasma membrane, then glucose entered, right? But that mechanism does not happen in all the tissues. That happens in the muscle. But in the liver, it doesn't happen. Liver, already those proteins are in the membrane. So now, one other terminology here I'll introduce, which is called isozymes. So what are isozymes? So different forms of an enzyme. So they will do the same reaction, but their affinity for the substrate is vastly different, can be vastly different. Okay? So it turns out hexokinase is one such example where there are 4, not 1, not 2, not 3, 4 different types of isozymes, isoforms. 1 through 3, they are found in the muscle normally. But hexokinase-IV is only found in the liver. The Km for hexokinase 1 through 3 for glucose is very low, and that's about 0.1 millimolar. At hexokinase-IV, Km really high, 10 millimolar. Hexokinase 1 and 3 are inhibited by G6P, but not 4. Based on these Km values, what do you think what it means? Why don't you guys talk among yourself for just about a minute, and then we'll discuss what do you think these different Km values means? Go ahead, Robert. Okay, that's all good. But the question still is why muscle hexokinases have such low Km, and why does so? What you are saying is that the muscle has a high need of ATP than liver. Is that true? It must be true, right? Muscle needs a lot of ATP. What it means, whenever there is glucose, muscle wants it first because muscle contraction needs a lot of ATP. So if you have a small amount of glucose, it will go to muscle first. Glycolysis, remember, is also happening in the liver, but muscle needs to do glycolysis first. Glucose will go to the muscle, and hexokinase 1 through 3 will trap it, make G6P do glycolysis. Only when the blood glucose level is high enough and the need for the muscle has been met, then only liver will take glucose. So normally after eating meal, our blood glucose is very high. Of course, first glucose will go to the muscle, and when muscle is satisfied, then only glucose will enter liver. If you think about the reverse, which is when you are making glucose in the liver, in what process? Gluconeogenesis. Liver is not doing gluconeogenesis to meet its own need. It is doing so that it can export to other tissues. We don't have much glucose under gluconeogenic condition. So whatever is being made, liver does not use it for its own purpose. It will transport to the muscle so the muscle can take it. Let's predict how would the Km look like, our MM plot. So if this is your velocity and that's your glucose concentration, So it looks like that actually. So Vmax is low, but the Km is also very low, so it's fully saturated. And on the other hand, if you have, this is how liver will look like. So this will be about 10 millimolar Km, right? And this will be your very low, so about 0.1 millimolar Km. So next thing is the G6P inhibition. What does it mean that G6P inhibits 1 through 3 but not 4? 1 through 3 inhibited meaning that in the liver, what liver is saying that once a lot of G6P has been made in the muscle, right, then that means you're not doing glycolysis at a rate at which the G6P is being made, then let's don't make any more G6P. Let's send whatever leftover glucose to now liver, right? And so basically liver can continue uptaking glucose without being inhibited, right? Because muscles need has been made already. So now liver can continue making G6P without being inhibited. Make sense? And we need G6P to make glycogen, as he said, Thomas said, right? So the excess glucose can be stored in the liver to make glycogen. Okay, awesome. So now there is a mechanism by which this happens, which we'll look at next. So glucose enters liver cell through GLUT2, and this is already in the membrane, so there is no business of fusion. So once glucose enters, then it will do G6P, then H6P, then go on to do glycolysis, right? That's a normal pathway. But the activity of Hexokinase-IV is regulated by either keeping it in the cytosol or isolating it into the nucleus. If it is in the cytosol, it will make G6P. If it is in the nucleus, it will not. And there is a regulatory protein in the nucleus which controls its activity. So once it is in the nucleus, then it becomes phosphorylated. So again, we don't need to know all the details, but it will be associated with the regulatory protein. And remember, glycolysis is happening in the cytosol. So if the enzyme itself is not present, then glucose will not go on to make G6P. So glucose will be transported into other tissues. Only when glucose is high, see that glucose, you see the arrow going this way? Only when glucose is high, then glucose will permeate through the nuclear pore and make hexokinase-IV dissociate from the regulatory protein, and it will come back into the cytosol, make G6P. Now, when you are doing glycolysis, so your G6P going to F6P. So F6P competes for the same binding site where glucose will bind. And so F6P is not the substrate, obviously. So what F6P will do is that it will inhibit the activity of the enzyme, and it will promote its translocation back into the nucleus so that it is not present to make G6P. If you have generated enough F6P, that means the liver's need for glucose uptake has been made, right? Then isolate hexokinase and transport glucose into the muscle.